Well, welcome to the Image Data Science Interest Group webinar. We apologize for the technical difficulties, and I'm hoping many of you are still on to listen to the speaker. Today we have Dr. Greg Ferrell from Genentech. He's a distinguished scientist in the Department of Preclinical and Translational PK/PD and Translational Imaging. He provided a talk on data science and imaging in drug development from an industry perspective, being biotech and pharma. A few quick announcements. We do have the upcoming WMIC meeting coming up in Prague in September. Abstract submission is now open, so I encourage you to go to the website and submit your abstracts. We're trying to have IDS webinars monthly. Our next webinar will be March 22nd, and we'll focus on learning apparent diffusion coefficient maps from accelerated radial case sampling, diffusion-weighted MR in mice using CNN transformer. And the talk will be given by Yuming Li, a graduate student in the laboratory of Yong Fan at UPenn. And it will be moderated by none other than Dr. Joe Ackerman at WashU. So please mark your calendars for the webinar. Again, this webinar is given by the IDS interest group within the WMIS. The interest group is broadly seeks to advance the application of image data science across all research areas within the WMIS domain. I'm not going to go read the major topics of the interest group, given the time constraints, but I do encourage everybody that's interested to join the interest group, and I'm going to paste the link in the chat for anybody that's interested to join the interest group. A few notes about Greg. Greg received his PhD in biomedical engineering at UCLA, and following the PhD, he did a short postdoc before joining Genentech in 2008. At Genentech, he's currently a distinguished scientist, where his expertise in development and application of advanced modeling methods in drug discovery and development within the Department of the Preclinical and Translational PK/PD. He leads the Imaging, Modeling, and Simulation Group, which is focused on in part an analysis of in vivo imaging data using mechanistic and machine learning methods. He also serves as the MS, the modeling and simulation lead for the ophthalmology and neuroscience group within Genentech. And I am looking forward to his presentation. I'm going to stop sharing now, Greg, and you can share your screen.
Okay. One moment while I share my screen. Okay. All right, everyone should be able to see my slides. Thank you, Koresh, for the introduction. So today I thought I'd give kind of an overview of some of the various kind of modeling and simulation type projects that are ongoing at Genentech in support of our imaging efforts. So 4 topics will be touched upon today. I'll give a brief introduction into my modeling and simulation group here at Genentech, and then I will cover the highlights from 3 relatively recent projects that we're working on here involving dually labeled antibodies and cell uptake, tracer kinetic modeling in support of our CD8 PET program. And then we'll switch gears a bit and talk about artificial intelligence, deep learning-based approaches to automated image segmentation for micro-CT and PET, if there's time. So the imaging, modeling, and simulation group, I mean, you can read kind of the mission statement there, but essentially we provide modeling and simulation and advanced analytics support to a number of the core imaging groups at Genentech, both in the preclinical and the clinical space. Our key areas of interest, number one, kind of really our core skill set revolves around modeling and simulation and imaging tracer kinetics, PK/PD modeling, systems modeling, and this kind of thing. My group sits within the larger systems modeling group within development sciences at Genentech. And so within that group, there's a lot of expertise in this area. People typically come in with engineering backgrounds and have a lot of experience with modeling systems, differential equations, things along those lines. Of course, AI and machine learning have become huge in image processing and image analysis in recent years. So we've shifted into that area a bit. There are whole departments at Genentech and Roche that hire card-carrying data scientists that are exclusively focused on machine learning and AI for image analysis. But we've done some nice work, in my opinion, in this area as well, in collaboration with these other groups. And we're also driving some of the kind of more advanced mechanistic modeling efforts within our own department. For example, using quantitative or mechanistic modeling approaches to better understand tumor penetration of various antibody formats, for example. So we are interested in these areas, but equally important within this industry environment is when appropriate and when it's helpful, we focus on deploying models and tools that we develop. So for example, we use this IPACS data management and data analysis system. So this is a whole separate work stream that we have in our group, basically just to streamline data analysis and data handling, data processing. So if you pull images off of a PET CT scanner or a SPECT CT scanner, which we have in our own group, as much as is possible, we're automating the transfer to a central data repository. Within that repository, there can be kinetic modeling algorithms, machine learning or AI-based tissue segmentation algorithms that can be deployed at the push of a button, and it makes these tools very accessible to the typical imaging scientist. And it's all currently under development now. So the first example will be focused on modeling of tumor cell uptake of duly labeled antibodies. So this is a modeling analysis of in vitro time course data, and the applications are to our bispecific antibody programs and also to SPECT imaging. And so first, just a quick slide about bispecific antibodies. This is a major area of interest at Roche and Genentech. And these are, of course, antibodies where you have one arm that targets, for example, CD3 on a T cell. That will be your effector cell in this little panel down here. And then the other arm of the antibody would target a tumor antigen. In the example here, it targets HER2. So this is a way to really localize your treatment to tissues of interest, because in order to activate the T cells, the antibody needs to be bound both to the tumor antigen and to the effector cell, which in turn activates T cells and increases tumor killing. So the question here is that you have a change in valency. So you're no longer dealing with a bivalent anti-HER2 antibody, and of course HER2 is a breast cancer antigen, right? So you don't have both arms of the antibody binding HER2, so you lose that avidity component when it comes to binding. So your effective affinity of that single arm is less than if you had both arms binding to HER2. So the question is, when you change that valency from bi to monovalent, how does that impact binding to HER2, and how does that impact subsequent internalization and catabolism kinetics of the antibody? So our investigative pharmacokinetics group in my home department in development sciences, they've been running studies, initially in vitro studies in a KPL4 tumor cell line, where we label mono and bivalent anti-HER2 antibodies with both iodine-125 and indium-111. And so the key thing to take away from this is that I-125 is what we call a non-residualizing radionuclide, where once the radiolabeled antibody is internalized within the cell and catabolized, the free iodine leaves the cell essentially very rapidly. So when you're measuring the total amount of radiolabel in your in vitro study, that reflects, for the most part, I-125 that's associated with cell surface bound antibody. On the other hand, for indium-111 labeled antibodies, once that indium-111 antibody is internalized, catabolized, that indium label is trapped within the cell, primarily due to charge on that small fragment. So what that results in is accumulation of the label over time. It's not, I mean, some of it can escape the cell, but for the most part, what you get is like an integral over time. It shows the total cumulative amount of antibody that's been internalized by the cell, plus how much is bound to the cell surface. So this can be modeled. And so this is a high level schematic on the right of the compartmental model that we developed to describe this data. So again, as I mentioned, we have both the, maybe a little hard to read, but we have both the monovalent, one arm is HER2, the other is anti-GD, which binds to nothing. And the other antibody is bivalent for HER2. And we have both of these antibodies labeled not either with I-125 or indium-111. Everything is put into the plate simultaneously. And then so we can generate a time course of what is the I-125 and indium-111 signal over 3 days, say, in this in vitro system. And so in the model here, really the key things to take away from this is in the media, we model, of course, binding and dissociation to HER2, which is this kind of hook-shaped receptor here. Internalization of the antibody HER2 complex, then equations 8 and 9, and binding and dissociation, right, within the endosome, and catabolism of these antibodies, and then trapping here of the indium-111 radionuclides. So the reason we did this is that initially when the data was collected, and on the left, you can see the time course data for the monovalent GD HER2 antibody at the top and the bivalent antibody at the bottom. Just by visual inspection, this data is difficult to interpret. I mean, you can reasonably assume that, well, the bivalent antibody has a higher effective affinity. So your iodine 125 signal here, which is cell surface bound, is understandably higher. And then you get more internalization, more degradation, as reflected by the difference between the indium and iodine signals. Remember, indium gives you cell surface bound plus catabolized. Iodine is just cell surface bound. So you subtract the two and you get the amount of catabolized antibody. So you can't really say confidently just by visual inspection that this is all due to differences in affinity. So we used the model that I showed on the previous slide and evaluated different hypotheses. The hypotheses we evaluated, I'll go back, are that like the base model would say that when the bivalent antibody is bound to HER2, there's no change in rates of internalization, there's no change in rates of trafficking. And then we looked at all different other combinations where you have increased internalization of the bivalent antibody, increased rates of catabolism, and so on. So anyways, we looked at a bunch of different scenarios. The model was fitted, importantly, simultaneously to all the data that you see here. And the model fits are shown in blue. And the base model fits, which assume that that bivalent binding of HER2 has no impact on cell trafficking kinetics, is in gray. And so the base model cannot describe the data satisfactorily. You have to invoke some additional mechanism, such as increased internalization of HER2, which captures the data quite well, as shown in the blue slides. So the conclusion from this paper, based on this data, is that the differences in trafficking and residualization kinetics between the mono and bivalent antibodies are both due to binding and affinity, but also increases in the rates of internalization and degradation of the bivalent HER2 antibody. And we also have SPECT data using the same approach where we simultaneously inject these mono and bivalent antibodies labeled with I-125 and indium-111. So some of this was published in the paper where the modeling is described, but we're in the process of running additional experiments to support the translation of that tracer kinetic model from the in vitro system to the in vivo system. So I think that's the last slide for this section. So the next section, I'll focus on tracer kinetic modeling of CD8 PET data. And so the core objective here is to develop tracer kinetic models to calculate changes in CD8 concentration over time, especially changes in CD8 concentration clinically on treatment. These are patients, these are cancer patients who are undergoing cancer immunotherapy. I'll get a little bit into that in the next slides. So this PET tracer program, and there's a number of these CD8 PET tracer programs ongoing, as many of you are likely aware, but the motivation for this is that these PET tracers are used in conjunction with cancer immunotherapy. For example, drugs like atezolizumab that target PD-L1, essentially what it does is it increases the visibility of tumors to T cells and triggers a T cell response against the tumor. This image is frequently shown. This is sort of the cycle of the cancer immunotherapy response. I won't go through this step by step, but the data here is, I think, a little more intuitive to understand the motivation. So this is a commonly cited paper from Nature in 2014, and this and other studies like it were the motivating factors, in part behind this approach. And what we see is, and these are clinical tissue samples taken from melanoma patients. On the left, you have these 24 patients that progressed. In other words, they didn't respond to therapy. And these are all patients that were undergoing cancer checkpoint inhibitor therapy to activate T cells. And you can see what the difference is between these two patient groups. On the left, the patients that don't respond to therapy. And on the right, the patients that do respond to therapy, is that at pre-treatment, on the left-hand panel here, you see very low T cell concentrations, which is the brown staining, right? And then on treatment, you see, again, you don't see a significant increase in T cell concentrations, whereas in the patients that respond at baseline, you see a relatively high concentration of T cells in the margin, and then on treatment, infiltration of T cells into the tumor. So we want to develop PET tracers to be able to image this response within the whole body, because the current standard is just to take biopsies. And so, of course, this is a very limited, this is a very small window into what's happening into the entire patient. You just have to, the physician has to focus on a lesion that's actually accessible, and then it's just a small part of that lesion. And so given what we know about heterogeneity in cancer in general, that doesn't give a very, that's not very representative in terms of how all the tumors are responding in the patient. And then recently, late last year, our one-armed antibody tracer, the first batch of clinical data was published in Nature Medicine. This was a collaboration between Genentech and UMCG in the Netherlands. So in terms of the modeling, where the modeling comes in, and something to keep in mind with these kinds of tracers is we are not there's many different tracers that can generate a nice image, right, where the tumor kind of lights up. But really what we want to be able to quantify is the change in the PET signal, or the change in T cell levels in the same lesion pre-treatment versus post-treatment, because that's really what we're trying to evaluate. Did the patient respond to treatment? I mean, the baseline scans are informative as well, but really the critical thing is to see how have T cell levels increased in the tumors, right? So these are just a few images from the one-armed CD8 PET paper. There were a couple of arms in the study. This was the smaller of the two, and this was just establishing kind of the optimal imaging time point. So this was a patient with lung mets. And so that we settled on 2 days post-dose, post-administration of the tracer to image the patient. So the patient has to wait 2 days after being given the tracer. And then on the left are just some representative images from the publication. This study included patients with all sorts of cancers, right? A wide range of different tumor types. So the tracer indeed does indicate where you have high concentrations of CD8. And there was limited information on how pre-treatment versus post-treatment looks in these patients. Here's one example where this patient has a bladder lesion. Pre-treatment, your CD8 signal is very low here on the left. And then post-treatment, this really lights up significantly. So in our minds in the modeling group, we're focused on trying to quantify these differences. So sort of as a bit of an aside, but relevant to this is that different tracer formats will essentially give you different tumor to blood ratios or different signal to noise ratios at your imaging time point. So here are 3 tracers that we evaluated internally. On the left, I didn't unblind these, but we can do that. The one on the left is our one-armed antibody, where you can see your tumor to blood ratio at 5 days post-injection in a mouse is quite high. In the image here, you can see we have a non-binding control, and you can just barely see the tumor here. That's just non-specific distribution at that 5-day time point because your blood concentration is still quite high at 5 days. But then for the CD8 binding tracer, you see the tumor light up significantly, of course, since it's binding to CD8 in those tumors. On the other hand, on the right, this is our VHH tracer. It's actually closer to 15 kilodaltons in molecular weight. So it's a small antibody fragment, small camelid antibody fragment. And you can see here your signal to noise ratio is much higher, where this is at 90 minutes after injection of the tracer. For the non-binding control VHH tracer, your tumor signal is extremely low since most of it is cleared from blood, right? So your background signal is mostly gone. But for the CD8 binding tracer, your signal to noise ratio is quite high, as you can see here, and the corresponding dynamic curves are shown here. And then for a 75 kilodalton tracer, it's somewhere in between. You have a better signal to noise ratio than the one-armed antibody and a more favorable imaging time point at 3 days post-injection instead of 5. So we set out to quantify all of this and to try to gain insight into the kinetics and into how basically the utility of these probes vary in terms of imaging time point versus what would your signal to background ratio be in the image, and how does that impact your ability to actually estimate T cell concentration or CD8 concentration. So this is a tracer kinetic model that we developed for our kind of lower molecular weight, still a biologic, but closer to 15 kilodaltons in molecular weight. This is our tracer kinetic model that we use to describe that. It's fairly straightforward. The white compartments are just central blood, peripheral tissues. And then the tumor is split up into 2 kind of pools of compartments, just because that's what we see in the data. So that this is all kind of empirical. We see slightly more complicated dynamics compared to what we see with an intact antibody or a one-armed antibody. But what we can do with a model like this is we have this really nice mouse system where the mouse has a tumor xenograph that expresses, of course, some tumor marker. And then kind of at baseline, before doing anything to the animal, we do back-to-back scans of a non-binding control, right? And that would be the red curves here. So the tumor curve is the lowest one, and the blood one is kind of, you can see that labeled blood. But then we inject the animal with the T-cell dependent bispecific. So this is a antibody that binds to mouse CD3 and then to a mouse tumor antigen. And we wait at 4 days, scan the animal again. Again, we do these more or less back-to-back scans of the non-binding control and then the CD8 binder. And so you can see the blood curves lay really right on top of each other. The non-binding control in red looks very similar to the tumor uptake curve prior to treatment with the TDB. But since in those 4 days, you've had significant T-cell infiltration, you get very high uptake in tumor binding. So what we do is we take this model here and we simultaneously fit it, like for each of the panels, we simultaneously fit that model to all of this data, right? And when we do that, we can get a fairly accurate estimate of CD8 concentration. This Bmax is CD8 concentration in the tumor. So we get an estimate of about 0.1 nanomolar prior to TDB treatment, and then close to 2 nanomolar after 4 days after administration of the T-cell dependent bispecific, right? So, okay, how do we know those values are accurate? So what we did is, so what I've described so far are how we calculate the CD8 concentrations at T equals 0, which is prior to the administration of the T-cell dependent bispecific. This is that average here in 4 mice of about 0.1 nanomolar. And then 4 days later, we use the tracer kinetic model again, and we get this estimate here closer to 2 nanomolar. And then on day 5, so okay, we have time points, day 0, day 4, and then on day 5, the animals are sacrificed, the whole tumor is excised, and then we use FACS to generate an estimate of CD8 concentration within that tumor. And as you can see here, it is, qualitatively looking at this, it is consistent with the estimate that we calculated using the tracer kinetic model at day 4. So this gives us some confidence in this approach, and that we can generate accurate estimates of target concentration before and after treatment in this mouse model. And this is, of course, using a non-binding control in conjunction with the CD8 binder. But this kind of, shows, if you set the bar very high, this shows like how much information you can actually extract from PET scans if you have the right data. So then we took this a step further, and using the same modeling approach, the question we were asking, a while back was, what at a high level, how can we expect a 15 kilodalton tracer? This is the VHH here on the left. How can we expect that tracer to perform relative to our one-armed antibody, which is closer to 100 kilodaltons? So what we can do, and we had dynamic PET data from many mouse experiments. And so that model that I showed previously was fitted or calibrated to all of this data simultaneously. And then we can generate curves like this, which show theoretically, given all the assumptions, which there are always a lot of assumptions in mathematical models, but given all those assumptions, we can generate an approximation of how the PET signal in tumor will change along with the CD8 concentration in the tumor. And when you do this, kind of head to head and compare two different tracer types, it gives you some insight into the utility of the tracer. So not surprisingly, what you get is that for the lower molecular weight tracer, at a 3-hour, this is in mouse, at a 3-hour post-injection time point, you have a much wider dynamic range compared to the one-armed antibody at 5 days post-injection. And this is due to two things. One is the affinity of the tracer is about 10 times higher for the VHH, but also the PK, pharmacokinetics, is much faster, as we would say. Basically, the free tracer clears very rapidly from the animal. And so within a matter of a few hours, you're left with mostly tracer that's bound to CD8 in the tumor. So you can base, this is on a log scale here, so I did not show the values, but this can span 2 orders of magnitude in terms of its dynamic range, being able to actually detect changes in CD8 concentration, whereas the one-armed antibody tracer spans about 1 order of magnitude. So the apparent benefits based on this analysis of the VHH, the low molecular weight tracer, is that there's a lower detection threshold, right? We can detect these so-called immune desert conditions, which that's like going back to the very first slide that showed the tissue biopsy, where you have very little T cells in the tumor. You have a greater chance of detecting those conditions. Higher dynamic range, as I just described. And then, of course, that higher dynamic range is due to a lower background since the tracer clears more rapidly. So these are all things that we know, right? I mean, these are characteristics of the tracers that are very well understood. But this quantifies that, which is very helpful. So when we come to like decision points in these programs of what's the advantage of moving forward with a program like VHH? This is very helpful in framing those discussions and putting some actual numbers behind these concepts that we talk about. So some of the advantages of the higher molecular weight tracer on the right is that you should still be able to differentiate between high CD8 levels versus moderate CD8 levels. And this is likely due more to the lower affinity of this one-armed antibody tracer, but the dynamic range potentially extends into higher concentrations of CD8 versus the VHH, where this curve kind of plateaus at a bit of a lower value compared to the one-armed antibody. So in terms of all the mouse work that we did and discussions in moving these programs forward, this was sort of the culmination of all the modeling that I showed part of in the previous slides. So this gives some insight into how, what is the value of modeling? You can use it to actually quantify CD8 concentrations, target concentrations, but you can also take it a step further and generate simulations, like prospective simulations like this, that show what's the sensitivity of your PET signal to changes in CD8 concentration. And so this same model that I described, and just as an aside, this speaks to the deployment aspect of our group, right? When we develop tools we think are useful to other scientists, we like to kind of package them and make them actually accessible. So our modeling and simulation group at Genentech, Iraj Hosseini and colleagues, developed this MATLAB GUI, right? It's basically a customized interface to the modeling tools that are available in MATLAB Symbiology specifically. But so we created what we call a session file with this model that allows someone to actually use it via a GUI that pops up as basically one screen, where it makes it much easier to work with the model. You can load your data. Everything's driven by turning checkboxes on and off. It's got a run button, that kind of thing. So this just makes this modeling tool more accessible to people who are running imaging studies. So just a few more slides on this topic. So I really wanted to focus on the mouse work that we did, because I think that gets across just the key concepts and the key motivations of why we're doing this and what we think the value of modeling is in PET tracer development and PET data analysis. But we are taking it a few steps further. And I'm only going to show the model here. We're still in the process of writing up and publishing some of this work. But this is a model that was built for monkey dynamic PET scans, again with the VHH tracer. But now, instead of just having blood and tumor, we're expanding the model a bit. And it's based on this very classical model within the clinical pharmacology community in the drug development industry that came out of UCSF. Nick Holford and Lou Shiner are huge names in this area. So kind of using this as inspiration, we developed this more like PET tracer specific model where you represent, of course, blood, binding of the tracer to CD8 within the blood, kind of a lumped term here where you capture distribution to CD8 positive tissues, which will be lymphoid tissues in a healthy monkey, and then binding to those CD8 receptors, and then we explicitly represent peripheral tissues. And so we had a lot of success with this model, and this is forthcoming perhaps to be presented at an upcoming meeting. But that's kind of the next step in the model. Going beyond that, and what we're very actively working on now, is taking more of a so-called PBPK or physiologically based pharmacokinetic modeling approach to analyzing PET tracer data. So now we explicitly represent different CD8 or target positive tissues like spleen, bone marrow, lymph nodes, liver, kidney, lungs, GI, can be all lumped into one compartment and so on. I won't go into a whole lot of detail here, but now I think you see the pattern. We're going from just having blood and tumor, then blood, lymphoid tissues, peripheral tissues, and then here the culmination will be explicitly including all tissues of interest in the model. And this facilitates calculation of tissue-specific dosimetry, receptor occupancies. We can explicitly include hot and cold fractions of the tracer dose if that's needed. And then the big thing, of course, is this will be used to inform translation of PET tracer distribution and binding from animal to human, which is the key thing that we do, of course. Like when we collect PET data in monkeys and in mice, we need to make projections as to how the tracer will behave in a human, and a modeling framework like this will go a long ways towards that objective. Okay, so that wraps up the CD8 part. I have about 9 minutes left. I want to leave some time for questions. So the last part, which I'll go through a bit quicker. I see I have less than that actually. So this is I wanted to focus a bit on what we've done in automated tissue segmentation, right? So this is switching gears entirely, basically. But okay, so briefly, we have a lot of data at Genentech. We have a repository of about 3,000 micro CT scans of these genetically engineered mouse models for lung cancer. These are KRAS P53 mutant transgenic mice, where if you administer an adenovirus, then they begin to develop lung tumors. You can see some of them in these cross-sectional images here. But we have what we used as a training set of about 3,000 of these scans with manually drawn lung regions of interest. It's super time consuming to do this. It's bias and error prone, right? So the first question is, can we develop an automated AI approach to fully automate the drawing of these lung regions of interest? The answer is yes. I mean, that is fairly straightforward with a large training set like this. But more challenging question was, can we segment out the tumors in the lung space? And can we segment out the individual tumors in the lung space to facilitate tracking of the response of individual lesions to various treatments? That's the interest, is do different tumors in the same animal respond differently over time to treatment? Another thing I'll note is that this training set included many different types of animals in terms of the magnitude of the lung tumor burden. It went anywhere from no tumors to a very high tumor burden. And we also had a group of animals from a lung fibrosis model that we included as well. So there's a wide range in different disease types reflected in this training set. So I'm not going to go into detail here. We used a standard approach, I'll say that. We used a convolutional neural network. This is a so-called VNet that accepts three-dimensional CT images that you kind of saw the animation scroll through there. We used a 3D UNet, which is very similar to this, but essentially we trained the UNet on those 3,000 images. We had a holdout test set that the network never saw to evaluate the performance, and it worked quite well. The DICE score was about 92% or 0.92, which is quite high for efforts like this. So that worked well. The next step was to use that lung mask as a first step in the tumor segmentation part of this. So just going through, and this is all published in the manuscript cited down there. So the first thing we do is we isolate the lungs. Within that space, you have, for the most part, just tumors and blood vessels. We go through a standard series of image processing steps, watershed segmentation here, number 5. And then we end up with an object with a map that just has all the discrete objects within the lung space. And then we developed a customized labeling tool, and this was kind of the time-consuming part. So for all the scans in the training set, we went through and manually assigned each object that came out of the previous analysis a label of either tumor, blood vessel, or we don't know what it is, which is common when you have very small fragments. Sometimes it's not clear what tissue type it is. So we created that training set. Then we used a support vector machine approach to classify, machine learning-based classifier to predict what tissue type that tumor is. So I know, I apologize, I went through that very quickly. What we ended up with is a single fully automated pipeline where you can feed in a micro CT image or a batch of micro CT images. It goes through all these steps. It executes the convolutional neural network, does the lung segmentation, does all the morphological processing, applies the trained support vector machine. And so your output is an image like this, or it's information like this, where you have the blood vessels in orange segmented out, and then individual tumors, sorry, the blood vessels in yellow, excuse me, segmented out, and then the individual tumors in orange segmented out as well. And qualitatively, it works well. it's certainly a solid first step in segmenting out individual lesions. I can kind of go fairly quickly through here. The one thing to point out in this slide is that we have time course data, right? So this is a single animal at the top where we have 5 micro CT imaging time points. This was a control group, and then a second animal that had 6 imaging time points and was treated with an anti-VEGF antibody and carboplatin chemotherapy. And so you can see here, this is a very nice first approximation of being able to identify individual lesions, and the individual lesions are color coded here, and tracking them over time. I mean, some user interferences, or the user needs to do some manual steps here in order to really do this, but this is a solid first step in being able to easily track individual tumors over time. This was published recently. A tool like this is, in our environment, is very valuable. We need to process about 100 CT scans a week where the lungs need to be segmented. So the lung segmentation tool part of it, this top part here, is used to process about 100 scans a week, which is a huge time savings and increases the quality of the data, because we saw in the manually annotated images that there are errors, there is bias from reader to reader, and these are all well-known things. And we currently do not have an open intern position, but we may have one in the next year. I'm very interested in continuing this work, actually. So anyone who's interested, let me know, and I can communicate with you on that. Okay, that wraps up most of it. I'll quickly go through this. I wanted to show a little bit of the clinical imaging work that we do. I'll say the main things I want to get across is that advanced analytics and data science are huge at Roche and Genentech. It's a major area of investment. In the image processing computer vision side of things, we're heavily focused on digital pathology, analysis of retinal scans, PET, CT, MRI. I can show a few examples of FDG PET that came out of our clinical groups. They've developed tools that are meant to be used to assist radiologists, for example, in segmenting lung lesions, lymph node lesions, and non-Hodgkin's lymphoma and lung cancer patients. These are meant to be tools for rapid initial assessments of images. They've taken it a step further and developed similar tools to estimate whole body tumor burden and then relate that to an estimate of clinical outcome, these so-called enhanced risk stratification for lymphoma patients. Additional FDG PET work that's been done is automated tools to remove the heart and the bladder. And this is just to remove interference with the signal in lesions that may be very close to the heart or the bladder. So, and there's a number of other ongoing efforts as well. This is just a few I wanted to point out. I've touched on all these points, but I'm not going to go through these again. Basically, kinetic modeling is very useful in a number of contexts. AI approaches are increasingly being developed and implemented within our environment and are also very useful. But I think I will stop there. Of course, thanks to all my many collaborators. Here's a nice picture of Genentech, the Genentech Research Center. So thank you. I'll stop there.
Thank you, Greg, and thank you for accommodating the talk to fit the delay in starting the webinar. I have a few questions, but also there are a few questions in the chat. I'm gonna read them out. So the first question, Hong Yuli has 2 questions. First one is, how did you guys quantify percent ID per gram in blood? I'm guessing in the first set of slides you had.
Yeah, so typically we draw a heart ROI.
And there's no spillover from the myocardium. There's no uptake in the myocardium
We're looking at a number of methods. Like, I think the more ideal, ideally you would want to draw a region of interest in the left ventricle of the heart, right, which is typically the largest blood pool. There's one method is drawing an ROI around the whole heart and then applying Otsu's thresholding or something like that to try to isolate the blood signal. Other approaches we've looked at are, we actually have developed an AI machine learning approach to this, where trained on a data set of manually drawn heart ROIs, have an AI algorithm automatically place a small sphere in the heart and then move that sphere around and kind of search for like a hot spot that has characteristics that are consistent with what you'd expect from a blood pool, which is like a high percent ID per gram and low voxel-to-voxel variability within that signal. But those typically line up pretty well when you compare them to blood sample-based measurements.
The second question here is in the lung segmentation, did you find inclusion of the bronchi to be beneficial to the nodule segmentation process, or did you opt to exclude bronchi?
So after we do all the thresholding and all the image processing, all that's left in the lung are blood vessels and tumors. The bronchi don't factor into it. They don't show up as tissue objects. So we do the initial thresholding just to remove everything except blood vessels and tumors.
Yeah, but lung tissue could have other cells, inflammatory cells, and so forth. So how do you distinguish lung tumor nodules versus other cells?
So that's a good question, and I sort of alluded to this, and especially when you're dealing with very small tissue objects, that can be challenging, right? For large tissue fragments, you can see very clearly when you're doing the manual labeling whether or not that's part of a blood vessel or if that's a lesion. For very small fragments, unless they're obviously associated with a larger tissue type that is known, it can be challenging. But in terms of the overall mass of tissue in the lung space, most of it can be accurately classified by the classifier. But for really small tissue fragments, that's a fair point. There is a sort of a size limit below which we start to lose confidence in the ability to predict what tissue type it is. Yeah.
Do you guys use gating for CT when you do this pipeline?
Yes, the tumor segmentation training set was respiratory gated data.
And do you see a difference between gating and non-gating? If you have that data, I'm just
curious. Oh, so that's interesting, and that's covered in the paper. But the model was trained on gated data, and then the big data set that we have, these 3,000 images, is all non-gated. But we applied that to the non-gated data, right? Both the images from both scanners were reconstructed to be equivalent. So we applied it to that, and it worked. It seemed to work reasonably well. Undoubtedly, it's not going to work as well as with the gated images, but just as a tool for a researcher to use to get an initial segmentation of individual lung tumors, it works qualitatively quite well.
And is this tool out? Is it publicly available, or is it?
That's a good question. Yes. It's all been published.
Wonderful, great.
If you're interested in the data or the tools, it's available on GitHub or contact me.
Wonderful, okay. I think we have two questions. I do have a question maybe early on with the modeling effort. Modeling is dear to my heart, and I'm sure to your heart. But as the models gain complexity, the question is, how do you get the parameter values for these complex models? There's so much you can fit data. So one question is, again, how do you derive the parameters for these complex models you have? And
so that you hit on sort of a key aspect of the broader systems modeling group that I'm in. So if the model is simple and you have the right data, like all the parameters are identifiable, you can just fit them to the data.
Right.
As models get bigger, you have more uncertainty maybe in the structure of the model and the parameters themselves. You lose that identifiability. We take what's called a virtual population approach. This has been published. There is a MATLAB toolbox that our group published. It's called GQSP Sim. You do a brute force grid search of the entire parameter space in a high performance parallel processing environment. You only keep simulations that are consistent with upper and lower bounds on the data that you've specified. But this approach works very well. It's the standard approach in systems modeling, and that's how you get around parameter identifiability issues. We've done head-to-head comparisons between actual parameter estimation and virtual population approaches. It's a very interesting area, but this virtual population approach is like the cutting edge of systems modeling in the drug development industry and in some parts of academia as well.
Well, yeah, I think you're on the mark. Greg, again, thank you for your time and braving through the technical difficulties and working through your presentation. And I want to thank everybody that stuck around, given the delay. Again, thank you so much. And join the interest group if you're interested. Greg is a member of the interest group.
That's right.
We'll be happy to engage in questions offline.
Yeah, thank you, Koresh. Thank you for the invitation. It was a pleasure, everybody. Thank you.
Bye, everybody.
All right, you are no longer live. I'm so sorry. It's like Zoom just quit.
It worked out. It worked out. What was that? How many people were online?